Alrighty, so this week's exploration, we'll be hearing from Arcadia, who is in cell biology, and she will be talking about how flatworms can be used to help understand stem cells. So I think that I will go ahead and stop sharing now and hand it over to Arcadia.
So hi everyone, I'm Arcadia, and I'm in my second year of grad school. And I'm excited to tell you today about how I study stem cells using these really cool worms that can regenerate. And as was mentioned, these are flatworms, so I'll be talking about a different category of invertebrates than the insects that we were just talking about. And before I get into talking about the science, I first wanted to just introduce a little bit about myself. So I'm from Central Massachusetts, and when I was a kid, I always thought science was pretty cool. I always really liked rocks and looking for fossils on the beach, looking at stars and learning about different constellations. But I specifically remember in 7th grade chemistry class, sorry, the slides are going a little bit slow. There we go. In 7th grade chemistry class, we learned about the periodic table of elements and all of these different elements that make up everything around us. And that's when I became really fascinated to think about what everything is made of and how it works. So I became really curious about everything around me and wondered, what are plants made of? How can they use the energy from the sun? What are stars made of? Why are they bright? Why can we see them in the night sky? So I had all these questions about everything around me and how everything worked. And then I also realized that the human body itself is a pretty interesting thing. How does it function? How can we taste? How can we see? How do our cells work? And so that's why I decided to study biochemistry in college. And I went to college in Western Massachusetts at Smith College. And now I'm at Yale studying molecular biology, which means that I study the molecules or the different components that make up living organisms and study how these molecules interact with each other to enable organisms to function. And then more specifically, I study stem cells. So I'm curious, what have you heard about stem cells? Do you know what stem cells are? Maybe you can type in the chat what you've heard about stem cells already. If anything, it's okay if you haven't heard of them. They're cells that turn into other cells. Yeah, that's a great suggestion. Yeah, so some people have and some people haven't heard of stem cells, and that's fine. But as someone suggested, stem cells are cells that can turn into other kinds of cells. So in our bodies, we have lots of different kinds of cells. So we have some skin cells, which provide a barrier between our bodies and the rest of the world. We have special cells in our eyes that can detect light, and that's what enables us to see. We have different types of cells in our intestines that can absorb nutrients from the food we eat. And stem cells are the cells that can turn into all these different kinds of specialized cells. So, why are stem cells important? If we think about a developing embryo, it first starts out as a single cell and it grows into a collection of more cells. And eventually this turns into all of the different tissues and that make up our organs and that make up our whole body. And this is lots of different kinds of cells. And the process of a stem cell turning into a specialized type of cell is called differentiation. So this is the process as a stem cell becomes more and more specialized and then eventually becomes one of those specific cell types like a skin cell or an intestinal cell. And so clearly stem cells are really important during development, but they're also important in adults. So even though we're done developing, we still need stem cells as adults because each of our cells has a set lifespan and eventually it will die. And we need stem cells to be able to replace our old cells with new cells. And we also need stem cells to be able to make new cells if we get hurt. So for example, if you scrape your skin, we need stem cells in order to replace the skin cells that we lost when our skin was injured. So this process of replacing cells and replenishing them is called regeneration. And if we look at this diagram here, you can see this is a cross-section looking at an injury to your skin. So let's say a paper cut. You've probably all had paper cuts before, or scraped your knee or something. So just to orient you in the picture, the blue is outside the body, and then you can see the layer of skin cells on the edge, and it's broken here, and you can see it. There's a bit of blood, different kinds of blood cells in there, and then as you've probably seen, if you've ever had a scrape or a cut, it might bleed a little bit, and then eventually it will scab over, and then eventually the skin will reheal, and you'll have a new layer of skin there, and there won't be any cut anymore. So this is the process of healing, and regeneration is important to this process. So why do we study stem cells? Well, I study stem cells because I want to understand how things work. So I want to understand how a baby can grow from a single cell, or how we can heal ourselves after an injury. But beyond just being interesting, this information can also be really useful and can contribute to medicine. So there's a whole field called regenerative medicine that's trying to use stem cells to help people heal from injuries or illnesses. So for example, sometimes if people get sick or get hurt, they might need an organ transplant if one of their organs stops working. And if someone receives an organ from someone else, there's a chance that their body will reject the new organ because their body recognizes it as something foreign and not part of them. So people working in regenerative medicine want to use a person's own cells to grow them a new organ that their body won't reject. So this is one of the goals of regenerative medicine. And if any of you were here back in May, Chris gave a talk about how his lab is studying how to make new hearts. So if you weren't able to see that talk, I recommend you can go back and watch the recording of it online, because he gave a really interesting talk about how he uses stem cells in regenerative medicine. So Chris's regenerative medicine research is what we call applied science, because we're using science for a specific purpose, in this case for medicine. And my research, on the other hand, is what we call basic research. So I study the basic properties of stem cells in order to try to understand how they work. And the better we understand how stem cells work, the more easily we can then use them for purposes like in medicine. And this is also how we can discover new things that we didn't even know were interesting before. So just to give you an analogy to understand the difference between basic research and applied science, let's just think about baking for a minute, because most people like food, like bread and baked goods, right? So if we think about baking, baking a loaf of bread is kind of like applied science, where you're combining different known ingredients to make a loaf of bread, and you're following a recipe that you might change and develop depending on what kind of bread you want to make. But you know that if you combine, eggs and flour and yeast, then you'll get some bread. But in order to do that, someone first needed to figure out how to make flour from wheat and study the different properties of what happens when you combine different ingredients, how to use yeast to make the bread rise. So all of those basic information that you need before you can make the bread, that's kind of like the basic research. All right, so I study stem cells. How do I study stem cells? It's actually pretty difficult to study human stem cells since it's not ethical to conduct experiments on humans. We can only study human stem cells in culture, which means growing them in a petri dish in the lab, like in this picture. And then the bottom picture is a microscope image looking at some cells that are growing on the surface of a petri dish like this. So studying cells in culture can give us some important information, but cells also behave differently when they're part of a living organism. Because when a cell is in a tissue and interacting with other cells, it behaves differently. And for that reason, we like to use model organisms as well. So I'm wondering if any of you can name some common model organisms. Maybe if you've come to one of these talks before, you've heard people talk about other organisms that they study. I see someone said mice, that's a very common one. Rats and mice, and plants, some other kinds of worms, fruit flies. Yeah, so it sounds like a lot of you are very familiar with some of the common model organisms. So we have mice, zebrafish is another common one. I don't know if anyone said that. Fruit flies. We've had some other talks in this series about C. elegans, which is a kind of worm. And the organism that I study is a bit different from these. It's less common, but still a very useful model organism called Planaria. So Planaria are freshwater flatworms, which means they naturally live in freshwater streams and ponds. And this is a picture of one. So you can see it has a head end up here. You can see it has these two little eye spots. And the eye spots allow them to detect dark and light. So they can't really see complete pictures like we can, but they can just sense dark and light. And then the other end is called the tail end. And then this little oval in the middle is called the pharynx, and that's what they use to eat. And I'll talk a little bit more about that later. And this is a picture looking through a microscope, just to give you an idea of how big these are. This is a picture for scale. So this is my hand, and then these are some of the little dishes that we keep the worms in lab. And then you can see each of these little things is a single worm. So they're about the size of a grain of rice or a little bit smaller. Yeah, so these are Planaria, and these are a really, really cool organism. I think they're the coolest organism that exists because they are what we call biologically immortal. So I don't know if you've heard this term before, but it basically means that while they can die, if they're given the right conditions, they can just live indefinitely. So they don't age, they don't go through the aging process that most other organisms do. So like if you think about humans, right, we start out as babies and we grow into children and then young adults and then eventually elderly adults, and Planaria don't go through this process. Planarian that's been there for, a year versus 100 years is, you can't tell the difference. You can't tell how old they are. Because they don't age, they don't have a set lifespan, so they can live indefinitely. And because someone might ask, they can die. So for example, if you take them out of water for too long, they'll dry up and die in that way. But if they're in the right conditions, they can just keep on living. So they're super cool. And then I have a video that will hopefully work. It's just to show you what it looks like when it's swimming around in the water. And it's pretty cute, so I'm hoping that the video will work. It's usually a little bit slow. So we're going into the eyepiece, and then there's one of the Planaria just swimming around in the water. You can see its eyes. So hopefully the video is working for everybody and not too laggy. And then, yeah, so that's what the microscope looks like, and you can see that it was in that little dish like we saw before that we keep them in lab. And yeah, I think they're super cute. Their eyes make them look like cartoons. They don't even look real. All right, so since we study Planaria in lab, we have to feed them. So this is a picture of a Planarian eating. And it eats out of its pharynx, like I mentioned earlier, which is this straw-like structure. There's my mouse, there we go. So this here is the Planarian, and then this white tube sticking out, this is the pharynx. So it's basically like a straw that it can extend from its body and then use to suck in the food. And then the red stuff here, let's see. The red stuff here is the food that we're feeding it. And we've actually just dyed it red so that we can see if it ate or not. And because the worms are kind of brownish, but they're also kind of see-through. So after it eats, we can actually see through to the intestines to see the red food inside and see that this is one that just ate. So that's why we dye it red, just so that when we're working with the planaria, we can see which ones are eating to see it, make sure they're healthy. So this is one that just ate, so you can see right through it to see the red food in its intestines. And you can see the intestines make this kind of lacy shape throughout most of their bodies. All right, so I think we're going to do a breakout activity now where we talk about the different systems that the planaria have in their bodies. All right, so I hope that everyone had a chance to talk through some of these exercises a bit, and I don't know if anyone wanted to share the answers that you discussed in your group. Otherwise, I'll also just go through the answers. But hold on, let me reopen the chat to see what people are saying. So some people had a tough time with the kidneys. Yeah, that was a hard one. Yeah, I'll just start going through the answers. Number one, the digestive system was D. And it's two images here because I was showing in yellow, it's their intestines, which is kind of like what we saw in the previous slide where we were seeing the red food in their intestines. So their intestines make this, this structure that fills up a lot of their bodies. And then the green part here is actually the pharynx, which is part of their digestive system. Like I said, that's the part that they, it's kind of like a straw that they stick out and they use to eat. It's also where they excrete their waste. And then the next one, so the skin was B. In planaria, it's called the epidermis, but it's basically the same thing. It's a layer of cells on the exterior of their body that separates their body from the rest of the world. And then the nervous system was C. I think my group figured this out because there was sort of darker staining up here in the head end. So this is their brain up here. And then they have these two sort of central nerve cores that go throughout the length of the body. And then they have, you know, other nerves that connect throughout the rest. And then this other area here with concentrated nerves is actually the end of the pharynx, because since it's something that they stick out and sort of feel around for food, there's more nerves there. All right, and then the next one, the kidney-like system was A. So this one was difficult because instead of having two kidneys like we have, they have this whole series of kidney-like structures throughout their body that does a similar function where it's helping them to filter out. But yeah, that one was a little more difficult. And then the last one was muscle is E. So it doesn't really look like our muscles, how we have, you know, these different sets of muscles like our biceps and our hamstrings, but they just have muscle tissue that's sort of spread throughout their whole body that enables them to swim around and contract and stretch out a little bit. Yeah, but as you can see, they have all of these different systems that are kind of similar to some human systems. So even though they're pretty small, simple organisms, they're actually, you know, pretty useful for studying how stem cells can differentiate into different types of cells. And one of the discussion questions that you might have talked about in the breakout group was, what can planaria regenerate? So if they don't age, do you think that they can regenerate? Yes, and some people in the chat say everything, and that is correct. Planaria can regenerate any of their tissues, which is super cool because there aren't that many organisms that can do that. So for a human, if if a human has their head cut off, they're dead. There's no coming back from that. There's no regrowing the head. But planaria, you can cut off their head and they can regenerate their entire head. So cool. So here's a picture showing this is a planarian. And again, the head end is up here with the eyes. And then the pharynx is sort of in the middle here. And if you cut it in half, each half will grow into a whole new worm. So this is showing it takes about 10 days to regrow. So the half down here that lost a head will regrow the head, so you can see the eye spots starting to form, and eventually it's a whole new worm with a head and everything. And then the half that lost the tail and the pharynx can regrow the pharynx. This is showing the pharynx starting to regrow here, and eventually it has a complete tail and pharynx and it can eat again. And yeah, so it's super cool. And just so you don't think we're being too mean to them by cutting them in half in lab, this is actually something that they do themselves naturally. This is how they reproduce. They basically turn themselves into two, and each half becomes a whole new worm, and that's how they reproduce themselves. So they're super cool. And how can Planaria regenerate all of their tissues like this? It's because they have lots of stem cells, and specifically those pluripotent stem cells. So those are the stem cells that can turn into any kind of cell. Planaria have lots of these. So just to show you, here's some more microscopy images, kind of like the ones we saw in the breakout room. So in this one, we're staining for just all of the cells in their body. So all of the different kinds of cells are all stained here. And then in this second picture in green, this is staining for just the stem cells. So you can see that there are lots of stem cells sort of all throughout the body. And there aren't quite as many up in the brain area. There aren't any in their pharynx, but they're kind of in between all of the other tissues. And just to sort of illustrate that, this next picture in pink is staining for a bunch of their different specialized types of cells. So you can see that the eyes and the brain are stained, the intestines are stained, the pharynx is stained. Up here on the edge, you can see the epidermis, or their skin-like layer, is stained a bit. And then if we add these two pictures together, if we overlap them, you can see that the stem cells kind of fill in all the gaps between all their other kinds of cells and just sort of fill in all the extra space in their body. So they have this huge store of stem cells that enables them to regenerate like this. So then what I study is how can stem cells turn into all these other kinds of cells? This is the process of differentiation. And in order to understand how stem cells differentiate, we have to talk a little bit about DNA. So you may have heard of DNA, and it's essentially the instruction manual for the cell. So it provides the instructions telling the cell how to make all of the different molecules that it needs to function. So if we use an analogy, we can use an instruction manual to build furniture or build anything that you need in your apartment, for example. You can have instructions for how to build the walls that are the structure, how to build all of your furniture, instructions for how to put together your appliances like your dishwasher and your microwave and how they work. So just like that, the DNA has the instructions to make all of the different proteins in the cell. So proteins are the kind of molecule that do all those different functions in a cell. So there are structural proteins that help the cell keep its shape, and there are proteins that carry out all the different functions that a cell needs. For example, when we eat, we need our food to be converted into energy that the cells use. So there are specific proteins that carry out that function, and all of the other functions that are needed in the cell. And every single cell has the exact same instruction manual. So it's this really, really long, lots of different volumes instruction manual, really, really long strand of DNA, but different kinds of cells need to use different parts of the instructions, right? If you think about it, the cells in your eyes and the cells in your intestines have very different functions, and so they need different proteins to carry out those functions. So because of this, they need to use different parts of the instruction manual. So a big question in biology is how do cells know what parts of the DNA of that instruction manual they should access, and which parts do they not need and they can just ignore, and how does that work? So that's what I study. So the answer to this question is partly in the way that the DNA is packaged inside the cell. So in a cell, the DNA is stored in this compartment called the nucleus. And the nucleus you can think of as kind of like the library that stores all of those books. And the DNA is this really, really long strand, and it gets packed up around specific proteins and forms these structures called chromosomes, which you may have seen before. They're usually shown to look like X's. And then all this DNA is stored inside the nucleus of the cell. And in stem cells, all of the DNA is accessible. It's all sort of out on a bookshelf where the cell can look at all the different books that are there and decide which ones it wants. Whereas as the cell differentiates, so as it turns into a specific cell type, all of the parts of the DNA that that cell type does not need get packed up and aren't accessible by the cell. So it's kind of like all the books get packaged up into boxes and put in the corner, so you can't really access them anymore, and you can only use the ones that are left out on the bookshelf. So this at the bottom is showing what that looks like in DNA. So this black strand here represents the DNA, and the accessible DNA is open like this, and the cell can read it. And then the parts of the DNA that the cell doesn't need get packaged up tightly like this, and so they can't be accessed. So the cell knows to just ignore those parts. So I study this process, and specifically, I like to study what kinds of proteins are important in this process. So planaria are a really useful tool to study this, because if we're studying a particular protein, we can really easily see if it's important for the ability of stem cells to differentiate by seeing if the planaria are able to regenerate if we get rid of the protein. So here, if we're looking at a protein, let's just call it protein A, and we want to know if protein A is important for stem cells to regenerate, we can get rid of protein A and then see what happens to the planaria. So if we get rid of protein A and the worm, if we cut off its head and it's able to regrow a new head, just like normal, then we know that protein A was not important for stem cells to regenerate. But on the other hand, if we have a different protein, protein B, and we get rid of protein B, and then we do the same experiment, but if the planaria are not able to regenerate, they can't regrow their head, then we'll know that protein B was important for stem cells to regenerate. And then we'll know that we should investigate protein B more to understand what its role is in this process. And once we know all the different components that are important in this process, all these different proteins, then that will eventually help us understand better how stem cells regenerate. And then we can use this information in applications like regenerative medicine to help people figure out how we can regrow organs. And just to finish up, this is the lab that I work in, the Van Wolfswinkel lab. The head of the lab is Josine Van Wolfswinkel. And this is just a picture from one of our virtual Zoom lab meetings. So everyone in the lab studies planaria, but we all study different aspects of planaria. So I study stem cells and regeneration, but other people in the lab study aging, because since planaria don't age, they're a really interesting organism for us to understand why some organisms age and why some don't, and what that process is like. And some other people study DNA damage. So for example, if you're exposed to UV radiation, like if you get a sunburn or something, that can be bad for you and damage your DNA. So some people use planaria to study that. So planaria are this really, really cool organism that can regenerate and are biologically immortal. And they're also a really great tool for studying lots of different aspects of biology. So I'd be happy to answer any questions with whatever time we have left. And I hope you enjoyed learning about planaria.
Thanks so much, Arcadia. That was a great talk. I really feel like I learned a lot of information about planaria. Okay, so we have a bunch of questions, but I'm only gonna ask a few of them. The first, what process leads to differentiation of stem cells?
So there are multiple different scenarios in which a stem cell would differentiate. So, during development, stem cells regenerate into different cells. In adults, which is what we were mostly talking about with the planaria, the stem cells are activated by sensing a wound. So if the planaria, lose part of their body, the cells that are at the periphery there can sense that there's no longer cells next to them that are supposed to be there. And this activates a wound response and leads to the stem cells migrating from wherever they were nearby in the planarian. They migrate to that site of the wound, and then that's what initiates them to start differentiating.
Awesome. Another question, how many times can planaria regrow its body before dying?
So they can just keep on doing it. There's no limit that we know of. They seem to have this infinite ability to regenerate, which is what makes them super cool.
Awesome. And then a last question would be, how long does it take stem cells to become other cells?
That's a really good question. And I think it depends on the scenario. So if we're talking about planaria and we think about the fact that it takes them about 10 days to completely regenerate, it probably takes a few days for each part of that. So if you think about, but over 10 days, they can completely regenerate their whole new head or something. So each individual cell, somewhere within that 10-day process, is able to regenerate.
Awesome. I think Heather, do we have one more question? Have time for one more question?
Sure, why not?
Okay. A really interesting question is, how do stem cells decide what type of specialized cell to become?
Yeah, that's a really good question. That's part of what I study. So that process of how a stem cell becomes a specialized cell and how it knows which one to become and how it goes through that process is still not super well known, and that's why we're studying it. But part of it is, again, like sensing what's missing. So in planaria, if you cut off the head, it will be able to sense that that's what it's missing and know that it needs to regenerate like the brain and more nerve cells. Whereas if it loses the tail, it has it senses different cells are missing. And part of this is because it knows which part of its body is the head and the tail. And that's kind of another whole area of study that people use to study, that people use planaria to study also, is how an organism knows what part of its body is which.
Awesome. Thanks so much for answering our questions, Arcadia. Great talk.
Great, thank you.